Hi, my name is Lian and my group member is Chen Lan. We are going to introduce association of circulating tumor DNA testing before tissue diagnosis with time to treatment among patients with suspected advanced lung cancer. Our report includes introduction, methodology, results, and conclusion. In the first part, I will introduce the information of the chosen paper and explain what is liquid biopsy and plasma ctDNA genotyping, and also talk about the objective and question of the research. This is a study published in 2023 that focuses on the integration of liquid biopsy into the diagnostic algorithm for patients with suspected advanced non-small cell lung cancer, also known as NSCLC. Liquid biopsy is a simple blood test that detects tumor DNA in the blood, offering a less invasive and quicker alternative to traditional tissue biopsies. This method is especially useful when we need fast results to start treatment or when tissue samples are hard to obtain. Overall, liquid biopsy is a better choice than standard biopsy. It's a valuable tool for detecting tumor DNA in the blood. Plasma ctDNA genotyping is a specific type of liquid biopsy that focuses on identifying small fragments of tumor DNA circulating in the bloodstream. This method is particularly significant for NSCLC, as it provides effective, timely molecular profiling, helping to quickly guide targeted treatment decisions. The objective of the study was to evaluate the use of ctDNA genotyping before tissue diagnosis and its association with the time to treatment initiation among patients with suspected advanced NSCLC. The study aimed to determine whether ctDNA testing could expedite biomarker testing and reduce time to treatment compared to the standard tissue genotyping. And the primary research question was, can we improve the time to treatment using ctDNA genotyping before tissue diagnosis among patients with suspected advanced lung cancer?
In order to fully understand this paper and its technology, we will dive into the methodology part. The process of this clinical trial includes selecting patients, obtaining their consent, and then conducting plasma CT testing. Following this, patients undergo a diagnostic tumor tissue biopsy with reflex molecular testing. Additionally, a reference cohort undergoes standard tissue genotyping. And finally, we outline the study endpoint to compare diagnostic timelines and the efficacy of integrating ctDNA testing. This is the workflow of the methodology. After eligibility confirmations, patients were invited to participate in the studies, like in this figure. Consenting patients underwent a peripheral blood draw. Approximately 20 ml of blood was collected in straight tubes and shipped to the clinical laboratory for the analysis. The patient's diagnostic workup continued per institutional standard, including the mandatory diagnostic tumor tissue biopsy, and patients with confirmed non-small cell lung cancer had reflex molecular testing, including the tissue NGS for non-squamous non-small cell lung cancer. The plasma results were returned to the study team and reviewed with the patient. Patients with actionable alterations identified in plasma received the medical oncology assessment. Whenever possible, treatment initiation was delayed until the tumor biopsy were performed unless the patients were too unwell and needed to start treatment earlier. Now we will discuss in more detail about the plasma ctDNA testing. All consenting patients undergo the plasma CT testing, and the procedure included the blood collection, with 20 ml of blood is collected, and the testing method is the plasma ctDNA testing using the Envision First Lung NGS assay targeting 37 genes relevant to lung cancers. The processing and analysis include the blood sample were processed to plasma using the QIAM circulating nucleic acid kit. DNA were extracted, sequencing libraries were prepared, and sequenced using the Next Sequence 500 system, and data were analyzed using the Inivata analytical pipeline. Results were returned within a median of 7 days from blood collection. About the Envision First Lung NGS assays, it is the assay used to target and detect the specific genetic alterations in the patient's ctDNA, and the functions of this is to detect the genetic alterations in circulating tumor DNA from plasma. It targets the 37 genes relevant to lung cancers as represented in the figure. The purpose is to identify the genetic mutations, single nucleotide variants, fusion, and copy number alteration that can inform the treatment decisions. The advantages of this assays is the cost-effective, targeted NGS-focused sequencing effort on specific regions of interest and reducing the overall cost compared to the whole genome sequencing. And high sensitivity, it allows for detection of low-frequency mutation in ctDNA, which is critical for early cancer detection and monitoring, and for the clinical relevance. By targeting known cancer-related genes, targeted NGS provide clinically actionably enable information that can guide personalized treatment decisions. After that, all patients underwent a biopsy of the lung tissue to confirm that the patient has non-small cell lung cancer. Once the non-small cell lung cancer is confirmed, further testing of the same tissue sample is automatically initiated to look for specific genetic genes in the cancer cells. The procedures included comprehensive NGS using the uncombined comprehensive assays version 3 of Thermo Fisher Scientific, immunohistochemistry for tumor PD-L1 expression, and other types of mutations. And single gene testing is using for several of the genes like in the slide. The uncombined comprehensive assay version 3 is one type of the targeted next-generation sequencing, ion torrent semiconductor sequencing. Its key features including targeted sequencing, focus on a specific set of cancer-relevant genes, detects hotspots, SNVs, indels, CNVs, and gene fusions. The gene panel can comprehensive profiling of numerous cancer-related genes. The multiplexing is simultaneously sequence multiple genes in a single run, and the technology mechanism, it detects the hydrogen ion release during DNA polymerization, utilize the semiconductor chips for sequencing. Reference cohort provides a historical baseline for comparison. This cohort consisted of patients diagnosed with advanced non-small cell lung cancer and treated prior to the COVID-19 pandemic. By comparing the Accelerate cohort with this reference group, the study aims to demonstrate the effectiveness of the new plasma ctDNA testing approach in reducing the time to treatment and improving the identifications of actionable genomic alterations. The inclusion of the reference cohort helps to highlight the advancement made by integrating ctDNA testing into the diagnostic algorithm and provides a robust context for evaluating the study outcomes. The primary endpoint was the time from referral to treatment initiation among patients with advanced non-squamous non-small cell lung cancer using ctDNA testing before diagnosis compared to a reference cohort using standard tissue genotyping after diagnosis. The secondary endpoints include the frequency of actionable targets identified using the plasma ctDNA, time to sample collection, and the turnaround time for plasma versus tissue profiling. This methodology allows for a detailed comparison of the diagnostic timelines and the efficacy of integrating ctDNA testing into the initial diagnostic workup for patients with suspected advanced lung cancer.
I'll talk about the results of this research. All participants in the study underwent plasma testing. Out of 150 patients, 115 or 77% had tumor-associated variants detected. Focusing on the 90 patients with advanced NSCLC, we found that 46 patients, or 51%, had ESCAT Tier 1 actionable driver alternations. Additionally, 20 patients, or 22%, had non-actionable driver alternations. Finally, 24 patients, or 27%, have uninformative results. Among the 90 patients with advanced NSCLC, 74 patients, or 82%, had successful comprehensive NGS in tissue samples. Another 9 patients, or 10%, had sufficient tissue for single gene testing only, while 7 patients, or 8%, had insufficient tissue for any testing. In total, 42 patients, or 47%, had ESCAT Tier 1 actionable alternations identified in their tissue samples. Combining both plasma and tissue results, 53 of the 90 patients, or 59%, had actionable alternations identified by either method. Of these, 35 patients, or 66%, had actionable alternations detected in both plasma and tissue. 11 patients, or 21%, had alternations detected only in plasma, and 7 patients, or 13%, had alternations detected only in tissue. Most of the actionable alternations were identified in adenocarcinoma cases, with no ESCAT Tier 1 to 3 variants detected in squamous carcinoma cases. Most patients started systemic therapy, with a median time to treatment of 39 days in the accelerated cohort, compared to 62 days in the reference cohort. For those with actionable alternations, 75% began targeted therapy, with a median time to treatment of 33 days in the accelerated cohort versus 61 days in the reference cohort. Additionally, 23% of patients initiated targeted therapy based on ctDNA findings before tissue results, with a median time of 27 days. Combining plasma and tissue NGS results, the median time to treatment was 41 days. In the accelerated cohort, the median turnaround time for plasma testing was 7 days, significantly shorter than the 23 days for tissue NGS. The time from referral to blood draw was also shorter at 7 days compared to 12 days for tumor biopsy. The median time from lung referral to plasma DNA result was 15 days, while it was 35 days to the tumor tissue NGS result. Plasma NGS results were available 17 days before tissue NGS results. In the reference cohort, the median time to tissue biopsy was longer at 24 days compared to 12 days in the accelerated cohort. The median time from referral to molecular result reporting was 42 days in the reference cohort, slightly longer than the 35 days in the accelerated cohort. Finally, I will talk about the conclusion and further questions. The study concluded that plasma ctDNA testing before tissue diagnosis significantly accelerated the time to treatment and provided earlier identification of actionable genomic alterations compared to tissue testing alone. This approach has the potential to improve access to precision medicine and timely treatment initiation for patients with advanced NSCLC. Here are some further questions. How can we integrate ctDNA testing more broadly into clinical practice? And what are the long-term outcomes and cost effectiveness? And how can we improve the sensitivity and specificity of this method? These questions highlight the need for further research and potential refinements in clinical protocols. Thank you.